Hi science fans, I promised you that this lecture would be a little shorter so that I can both load it up on YouTube and also break up our unit a little bit more. So here we go, genetic modification, your online lecture. So let's talk about what genetic modification is to begin with. It is anytime we take genes, specifically genes, not just DNA, but genes, and move them either in between species, that would be interspecies modification, meaning we take something from a fish and we put it into a tomato, for instance, or intraspecies, and that would mean we see that there is an ideal gene for creating a lot of milk in a cow, and we see that there's another ideal gene in a different breed of cow that would produce milk at an earlier age because the female is reproductively mature at an earlier age. So an agricultural scientist might blend those two genes in the same species into the same cow so that we can get not only a cow that produces milk at an early age, but also produces a lot of milk. So that would be an example of intraspecies modification. We're going to focus primarily in this conversation on interspecies, the modification or the transfer of genes from one species to another species. And they don't have to be closely related because we all know that one gene is going to code for the same production of protein regardless of what species it's in. It's all in the DNA nucleotide pattern that codes for the amino acids, which produces the ultimate protein that does the thing that we want it to do. All right, so reasons for pursuing genetic modification. The number one is that somebody is attempting to improve on a species in some manner. And it might be gene therapy on human beings, where we're attempting to remove a gene that switches on to produce a cancer tumor, or it might be a replacement of a gene that doesn't actually produce a protein, a non-coding gene, and that's why the person experiences the disease or the trait that is undesirable. This is very limited still at this point, as opposed to agricultural modification, which is happening a lot. And in that case, you have an agricultural conglomerate or corporation that says, I think that I can produce a longer lasting banana getting it to market. And therefore, the customer, the consumer, is going to be more interested in buying my banana because it doesn't turn brown and blotchy as fast. So my banana will look more appealing, and therefore I will sell more of them. So it's worth spending all this money trying to identify a gene that will do that, transfer it, and grow it up. So an example of that you've heard a lot of is insulin-producing bacteria. So tricking bacteria into producing human insulin to be used to treat diabetes for anybody that has diabetes. And by having the bacteria produce the insulin, we're able to guarantee a regular, consistent supply of something that all humans are able to metabolize. So there's our classic example, and I know you've heard of it many times. I'm not going to belabor it. One you might not have heard of is the flavor saver. This is one of the original, rather early agricultural modifications that happened, and it was in a corporation's attempt to make sure that the time that it took for a tomato to be picked from the vine and get to market, there are times where that time period is so long that the tomato doesn't have the same high quality texture that the consumer is looking for, or the flavor has diminished because the tomato has aged past its peak. So one thing farmers can do is they can pick the tomato early, but another thing this corporation decided to do was to splice in a gene from something else, and I think it was a fish, but I can't remember at this point, not relevant. Splice in a gene from another species into the tomato to try to get that long-lasting tomato effect that they were looking for. They discontinued it after a couple of years because they found that overall, regardless of what point you were eating it, the actual flavor of the tomato was diminished. So and they also found that the firmness did not last at market. So it was an attempt to do it. It was not a successful attempt. But you know, scientists, they always come back for more. This is a fairly recent one that's being worked on, which is to splice DNA from a virus that would normally be used to create a vaccine, or rather, the virus could be used to create a vaccine by injecting it in a weakened state into a human, creating the human response to produce a protein. In this case, they're taking the gene that produces that protein, splicing it into a banana, the idea being that when people eat the banana, it will produce the protein that will protect against the vaccine, and over time, people would build up a recognition of that protein trigger and produce their own proteins, just like a vaccine. I don't think that we're there to market yet. My latest research suggests it's still a couple of years away. In some cases, people would want to grow faster growing trees. For them, that would be considered an improvement in an area that had been deforested from drought or from fire, or even from the situation we have here with palm trees having a certain life cycle. If we could grow them faster, then we wouldn't have empty, vacant land that was degrading quite as much. So, or if you were interested in harvesting trees for paper or for lumber, there's a lot of reasons why having a tree that grows faster and gets to maturity faster would be a benefit for commercial purposes. Having plants that are resistant to pests can do a couple of different things. People are working on this on a species-by-species basis. So if they can make it so that insects are not going to eat the crop, you have more excellent crop that you can get to market and more people that are able to buy it. It's also a potential way to reduce the spread of pests from one area to another. You've got bananas that are getting picked in South America and they're being transported to North America, and there are pest species that may be tucked away in the banana bunches and get a free ride to North America, where they find an environment with fewer predators or potentially new food sources, and they're able to thrive and be very successful. This would give an opportunity to decrease the spread of pests overall, as well as make it so that there are fewer resources needed to grow a plant up to maturity and get it to market. Let's see. Oh, and then of course there's also just the entertainment value of, we have a Kanga zebra antelope? Ibis? I'm not sure what those horns are. Mountain goat? Mountain ram? Anyway, this is obviously a photoshopped version of something, but there are certainly lots of organisms that we could easily swap DNA from one to the other. If nothing else, it would give scientists an opportunity to identify what some of these traits are able to do for the organism. In fact, for instance, recently, scientists have been debating whether or not the zebra stripes are actually a camouflage, or if they have another not identification purpose, but another survival purpose beyond camouflage. So this would give us an opportunity to determine if that's really the case. Although, of course, the predators on kangaroos and zebras are different. So I might just be rambling because it's a cool picture. Okay, so moving on. So we can use a couple of different vectors to transfer genes. Genes are small enough that we don't have currently really good tools to splice out a specific gene. But fortunately, we have some natural examples of this. And viruses are the sort of prototypical, or the one that a bunch of scientists started looking at and saying, hey, wait a minute, this is how it works. This virus is capable of inserting its DNA into a host. And we call them vectors because they are transitional areas or ways to transition DNA from one species to another. So viruses are really adept at doing this, and viruses are still used to transfer DNA from one species to another. You can cut out a little bit of the DNA, you can insert the desired DNA, and then you let the virus do its job, which normally is inserting DNA into a host's genome. They do it very well. We're going to focus primarily on bacteria. That's the example that the IB is most interested in you knowing. And I know you've heard the insulin example constantly. It is one of our earlier examples, and it's one of the, I think, least controversial examples of genetic modification. So to be able to transfer any gene into a bacteria, we are going to end up using the plasmids. And the plasmids, remember, are the smaller bits of DNA that are used for one bacteria to transfer to another one. This is their version of sexual reproduction that keeps the bacteria genome overall refreshed. It gives them an opportunity to exchange, get some new DNA to make sure that any mutations that are accumulating sort of get wiped out or reset, and it refreshes the diversity of bacteria overall, since ordinarily they're asexual reproducers or clones, and any antibiotics that are introduced into the community could, if they're all clones, wipe out the entire species or the entire population at one go. So these plasmids are a natural way for bacteria to exchange DNA and increase the diversity of their DNA. So we rely on the plasmids within the bacteria rather than the primary DNA that the bacteria is going to rely on to survive. There are restriction enzymes that are introduced into this process, and restriction enzymes are coded for a specific gene to recognize where in the plasmid do we need to cut in that DNA. And the restriction enzymes are specific for specific locations on the plasmid or specific genes. One of the things the restriction enzymes do is they create sticky ends, and the sticky ends will be a single helix section at each end of the gene that wants to get cut in, and it helps to attach the gene into the ends that have been cut out previously. And the reason it does that is the sticky ends have a single codon or a single DNA set of nucleotides that are looking for their complementary pair. And they find their complementary pair on the ends of the plasmid that have also been cut out. And that's how the DNA that is chosen to be added to the plasmid gets into the right place and stays attached, because it is actually bonded along the phosphate backbone. So the sticky ends are important for that. And then we see DNA ligase again, and I know that you've seen DNA ligase before, and if you remember before, its function was when we're looking at the lagging end of DNA replication in smoothing out the phosphate backbone. Hey, guess what? It does the exact same thing in the bacteria cell. So once we have our new DNA strand attached in here into the plasmid, the ligase comes along and just smooths out the phosphate backbone so that it is consistent with the rest of plasmid, and the bacteria doesn't recognize it as foreign material or an error to be corrected and cut that back out again. So once that has been entered into the plasmid, now every time this bacteria copies itself, it's going to be including that new piece of DNA that we've added in there. This is a way that we can get bacteria to make any protein we want, because again, any nucleotide strand in a DNA that is identical in a bacteria or a human or a cow, it's going to produce the same protein. So this is a way we can get bacteria to make us a lot of proteins for whatever function we need. So one of the things we're going to be relying on to get the information from the DNA source material is mRNA, because of course we know that our DNA is not going to function outside of the nucleus. It's just a blueprint. So we rely on mRNA to be able to transfer that DNA into just a small chunk of gene. We're going to rely on reverse transcriptase, because remember that the mRNA is not actually copying the gene, it's the complementary pairs. So reverse transcriptase is an enzyme that then produces something called cDNA, which is what we're actually going to insert into the into the plasmid in the bacteria. So just like with transcription and translation, there's a copying and a copy of the copy that is the chunk that we actually use. We have the same thing going on here when we create a new gene or a new copy of a gene to be able to insert into the plasmid. All right, so there's some upsides and some downsides. I'm going to kind of breeze through these pretty quickly. There are several reasons, or lots of reasons actually, to rely on genetic modification. Designing an efficient organism, this would be for commercial purposes. So if you're a farmer or you're raising cattle or sheep or even fish commercially, you're able to make an organism that actually produces denser meat or more milk, or gets mature younger, or has larger, meaty bits as opposed to bony bits. You can make sure that especially if you're doing agricultural crops like corn or tomatoes or potatoes, those are things that having a more appealing product means that your public is going to buy yours over somebody else's. As the water availability in the world changes, creating crops that have a less reliance or less demand for water is going to make a better product, according to everybody that knows about the financial side of the agriculture. Decreasing the use of pesticides and herbicides is a benefit not only because the farmer doesn't have to spend money on these things, but they also are not releasing as much chemical out into the environment once the crops that have been treated with pesticides or herbicides get rained on, and those chemicals run off into the streams and the rivers and the lakes and the oceans and all of that. So by decreasing the amount of chemicals that we are entering into the environment, we have the potential to improve the overall health of our environment. Having more reliability of crops and having crops that are less reliable on the variations of water or pesticides or need to provide fertilizers, if we can create crops that don't need some of these additives that are expensive or that may not be globally available, we can make it so that we can actually feed people everywhere in the world. Now, I don't know if you guys know this, we already produce enough food in this world to feed every single person on the planet. The problem is not the amount that we produce, it's where we're producing it and getting it to the people that are in areas that don't have as much water and don't have as much cropland. That's the challenge for us as a culture, as a community of humans on this planet. So there's a potential for the improvement or the use of genetic modification of organisms to make sure that we can actually grow stuff where people are hungry instead of growing it where people are full. So by decreasing the total amount of materials needed for growth, this means we can grow organisms in water and hydroponics by improving the receptivity, by improving the plants that we're actually trying to grow in the water and not relying on soil. Or we could require less fertilizer, or we could require less water. We could require different growing environments, which would make it potentially more effective for the farmer to grow. And then in some cases, we can change what we're putting into our food, therefore what we're putting into our body. So if we need to have foods that have a higher vitamin C content or foods that deliver more calcium, there are not that many foods that you can get protein from as it is, the kind of protein that we get from meat. And if you choose not to eat meat, you have to choose wisely what are the vegetables you're going to choose. There is potential to increase the protein content of leafy greens, for instance, or carrots. Something that somebody would want to eat that will also give them a nutrition that they might otherwise have a hard time getting access to. But there are some risks associated with this as well. And the number one is that pollen from any organism that is or plant that is genetically modified could potentially get blown or washed into wild crops, which would then compromise sort of our genetic bank, the area that we can go back to should our genetic modification not be successful. So it's important to keep some pure wild strains if genetic modification becomes a very widespread practice. We do end up with decreased diversity because right now we might have 20 different varieties of corn, for instance. If somebody hits upon the perfect combination of corn, we're going to see a lot less of the other less commercially perfect varieties of corn being grown. And again, we have a decreased diversity. That means if we have an environmental change or a pest that comes in that wipes out the entire population, it wipes out all the population because they are genetically similar and they are resistant or not resistant to the same things. So having a more diverse population means overall our habitat and our wildlife are safer from whatever might come through that would challenge them. Forest fire, drought, flood, pesticide, fungus, bacteria, things like that. Now, the other piece of thing that we need to consider with genetic modification is if you remove food sources for pests, you also are removing food sources for things that eat pests. So we're having right now a decline in populations of frogs all over the world, and one of the contributing factors is that we have decreased the presence of insects in the areas that frogs are living in, because we as humans like to avoid insects, or we don't want insects in our environment. They're super important for frogs and spiders and birds and a whole bunch of other things. So by removing the food sources or the places where they can live, we've ended up disrupting the whole system. Some species eat lots and lots of variety of things, and they can recuperate with some other species. Others have a much more narrow demand for their habitat. You might remember the word niche from your ecology section. And having a more narrow niche means that they are less resistant to the loss of a specific species or group of species to survive on. So that's a bummer. The other thing we can end up with, since I mentioned earlier that viruses are really good at inserting their DNA into other things, we can have genes that we have inserted into the population, the domestic crop population. We can have it jump into other species. And again, if you have a gene that is producing insulin, again, to use that example in bacteria, you put that same gene into a frog, that frog is going to produce insulin. It's the gene that produces the protein that is significant rather than the species it produces it in. But there are some species where it's going to cause them problems in survival, or you might give them traits that we don't want them to have, traits that make them more resistant to pesticides, for instance, if you are talking about insects, or fungicides if you're talking about a fungus. So instead of having a bread that is more able to resist mold, you might actually have given the mold the DNA to be able to resist the fungicides that would otherwise have killed it on the bread. So it's a little tricky to start controlling it once we get those genes out there. We can't guarantee where they're going to land. I think I mentioned already driving the pest evolution. I talked about pest eradication, but one of the things that can happen is you introduce plants that increase their resistance to pests. Some pests are going to survive, and those pests are going to be even harder to control or harder to kill because they are able to withstand the actions that the plants or the people that are protecting the plants are taking. And so we end up with more aggressive or intensive pests that are harder to control without adding larger amounts of chemicals into the environment. And we see this already happening with the use of pesticides or with the widespread use of antibiotics, for instance. We have now bacteria that are resistant to many commonly used types of antibiotics, and we have to dig deeper. All right, then we have some, I want to skip ahead a little bit here. This one here is regarding labeling of genetically modified food sources, because there are people that are pursuing specific dietary pathways, vegetarian, veganism, avoiding pork, avoiding red meat, and not knowing what's actually in their food can compromise their ability to pursue the life that they want, or they may have allergies to things. And if we've put peanut genes into tomatoes, you can have somebody having an allergic reaction without realizing what it was that they were eating. And then finally, there are corporations that are going to try to patent species because they've put a lot of money into creating this perfect combination corn, and now they're going to charge everybody a whole lot of money for it. So there's a concern there as well with access to food sources for farmers. So finally, at the end of all of this, you should be able to start thinking about whether or not allowing or supporting the expansion of genetically modified organisms needs to be a decision that a group of humans comes to and says, yes, we want to support this in our community because of these benefits, or no, we want to limit it in our community because of these concerns, these risks. And this is a decision that communities are working on all over the world. There's no one right answer. You know I love those questions that don't just have one right answer, but it's something for you to start considering as you're working through this unit. I hope this has been beneficial, and I will see you soon. Bye!